coming to our next speaker. Let's is Yael on? Is she on yet? Are you Yael? Dr. Weiss, are you on right now?
On, and I'm having trouble sharing my slides. It says someone else is sharing.
Okay, you have we have a few, one more minute because I'm going to introduce you first, and it says on the slide anyway, coming soon, so we are good.
Okay. You
So
set me
let
up.
me.
Thank you.
Yes. So let me introduce our next speaker. So where do we start? Right? Because there's a long career already, and maybe the latest. We start with the latest, and while Dr. Weiss is speaking to us. So Dr. Weiss founded Masi Therapeutics, a venture that is dedicated to delivering therapeutics to patients with underdiagnosed, and here's the new term, ultra rare genetic neurodevelopmental disorders. And I point this out because we had rare disease, we had uncommon diseases, we had nano rare, now we're talking about ultra rare. And I still hope in a few years we find, as I said many times now, there's commonalities between all the different diseases. Anyway, so we will also learn that later on what Masi Therapeutics, their approach, how to address all this. But before we come there, so I just want to mention also that Dr. Weiss has been having roles at Genzyme, Merck, Ultragenics. So this goes over 20 years back. So a long career here already. I also don't want to miss to say that she is a distinguished member of the NIH-driven Bespoke Gene Therapy Consortium, the ASGCT Translational Committee, N equals 1 collaborative, and holds the prestigious title of, and this is from 2022, Termere Fellow. The activity serves, she also serves as a board member for a number of organizations like ADNP and FOXG1 Foundation. The long career started with graduating from Hadassah, and I'm sorry for the pronunciation, medical school at the Hebrew University in Jerusalem, and she holds an MD and a PhD. And this will all lead us into her talk, revealing a little bit how Masi Therapeutics goes about developing gene-based therapeutics for genetic disorders. And I understand this will be, you will talk about your joint efforts that bring biotech, academia, and patient groups together. So looking forward to your talk, and the IT people here will make it happen that your slides will run properly. We can see you now. That's good news. And hopefully you can see us, and your slides should be up.
I'm just transitioning to slideshow.
Okay.
Great. So can you see the slideshow?
Yeah, it's not on presentation mode, but maybe there's, if you press it on presentation mode, we should be all good. To the right on the bottom. Here we.
No, just a sec.
Oh, this is you.
I dared to do it. Just a second.
We have you on the screen right now, and your slides are behind you. Can we help?
It's not moving to presentation.
Don't worry.
Let me try again.
We hear you well. We see you well.
Okay, tell me what you see now.
Maybe it's loading. It was there before.
I know. Here we go.
Here we go. Yep.
Great. So, hello, everyone. I apologize for not being there in person. One of the reasons that I'm not there in person is that this morning I was part of a meeting with the FDA, with the American Society of Cell and Gene Therapy, which we actually thought would not happen, which would have allowed me to come to San Diego in person, but it did happen. And even though Kent says that the regulators are slow and not in tune with what we're trying to do for uncommon or rare diseases, we actually had a really good meeting with them on the topic of gene therapies, and a lot of openness to creative trial designs, to creative approaches to endpoints. And basically, what the team that was on the phone told us was that we should talk to them as much as possible. So come to the FDA when you have ideas, ask for a type D meeting or a type B meeting or whatever meeting is appropriate for the question that you're planning to ask. And they will do as much as they can to accept your ideas and at least have a discussion on them. So that was really a good way to start the day with all the news that we're hearing about what's happening in the FDA. It was actually a very positive meeting. So Jeff set me up for a lot of the slides that I have, so I will go through them quickly, some of them quickly, not to repeat what Jeff's already said, but I do want to talk a little bit about the Mozzie perspective and how we approach working on therapies for, I know I'm adopting your terminology, uncommon diseases, and I would say very uncommon diseases, and a little bit about the lessons learned from what we've been doing over the last few years since Mozzie was started. So the key messages that I'd like to convey is that the more genes are being associated with genetic disorders. Obviously, we started with 7,000 a few years ago, and now we're over 10,000. The more genes there are, the more patient-specific and gene-specific organizations there are. And drug development in general, as Jeff said, there's a lot of failure. It's not for the faint of heart. I have a lot more white hair on my head now than I did when I started out on this journey. But we really, in the case of rare genetic diseases, I would argue that we have an advantage if we fully understand the biology, but it's not sufficient. It's not sufficient or not mandatory. And you can choose a modality that allows you to go ahead with the therapy, even if you don't fully understand the biology of the disease. For example, gene therapy, you need to know where you need to replace the protein, but you don't necessarily need to know exactly what the protein does. Proof of concept is only the beginning, and I'll talk a little bit about what we consider as proof of concept. And then why is it so important to work with advocacy groups? And why is this triangle of industry, advocacy groups, and academia so important? So the topics that I'll cover are a little bit about advances in genetic diagnostics. What is a target product profile? So when we go to investors, when we go to regulators, when we go to payers, we need to show what the drug that we're planning to develop is going to look like. What is it going to do? How many times a day is it going to be administered? What the toxicology profile of it is. There's actually a whole list of things that we need to address before we even start thinking about going into clinical trials. So I'll talk a little bit about that and then about the teamwork that's required to get there. So I talked about the number of genes. We work in the neurology and the neurogenetic space. So a lot of the examples that I'll be giving you are from this space. And for example, when you look at the number of genes that were known for epilepsy 15 years ago, there were very few genes that were strictly associated with a specific epilepsy clinical diagnosis. As you fast forward to today, there are hundreds of genes that are associated with epilepsies and very specific genes. And these could be channels, they could be secreted proteins, there could be nuclear proteins, transcription factors. There are many different types of proteins that actually, when they're mutated, lead to seizure disorders. And the genetic testing is key for many of these disorders that are just being associated with a gene, but it's key for commercial interests. So many companies are sitting back and saying, okay, we will wait till there are 10,000 patients and then we'll start developing a therapy. Other companies like Mozzie, we say, okay, we're going to start to developing a therapy, and hopefully by the time we get to the clinic and by the time we get a drug approved, there will be enough patients to make it commercially viable. So this is Mozzie in a nutshell. We're a small team. We're only 15 people, but all of us have a lot of expertise and experience with rare disease drug development. And the four bubbles, the four green bubbles exemplify our key partners in the journey to develop therapies. So we start with the advocacy groups. And this is where Mazi started, when I realized that a lot of advocacy groups, and some of them you heard from this morning, are developing therapies, spending money on getting proof of concept in cells or in an animal, and then kind of getting stuck because, as Jeff said, it's really expensive and it requires a lot of expertise. So we decided to start Mazi in order to be able to take programs that achieved proof of concept in an animal or in cell lines or in organoids and take these further into the clinic, take them into tox studies and into clinical studies. We work with the academics that actually conducted the work for the advocacy groups, because most advocacy groups look for the person that's an expert in their gene or in their protein and go to that person and ask them to start developing a therapy. So this is where the actual data will come from. And then we work with diagnostics companies because we're constantly looking for more patients for our disorders. And we work with CROs and CDMOs that execute a lot of the work that we're planning in order to get into the clinic. I'm giving SMA as an example. Jeff gave it too, but I'm trying to give it from a little bit of a different perspective. You heard about SMA from Jeff. It is a muscular atrophy, a spinal muscular atrophy. There's a gene mutation that's been known for a while. And until 10 years ago, there weren't any disease-modifying therapies for SMA. And currently, there's not only a small molecule, there's also an antisense oligonucleotide and a gene therapy. And these currently live together pretty well in harmony to give options to patients. And I think this should be the goal for every group and every gene, to be able to have a diverse set of tools to treat the disorder, because not everything, as Jeff said, will work for everyone. And kind of following up on what Jeff was showing, I have here the three different drugs that are available for SMA. The small molecule that was actually the last to be approved, the antisense that was approved in 2016, and the AAV gene therapy that was approved in 2019. So everything happened in the last 6 to 7 years. And you can see that there are big differences between them, and the mechanism complements each other, because the AAV gene therapy restores the protein by giving the missing gene. The ASO makes an alternative gene make protein, and the small molecule is a splicing modifier that also works on the genetic basis of the disorder. So all three of them work well. Price-wise, as Jeff indicated, the small molecule is likely the cheapest, and the AAV gene therapy is likely the most durable therapy of the three. So continuing on this line of comparing the small molecules to gene therapies and to ASOs, I highlighted a few things. I won't go through the whole table here, but I highlighted a few things that I thought were important. Small molecules, again, and Jeff, I'll constantly quote you. I hope it's okay. As Jeff said, they're the lowest hanging fruit if you're repurposing. They're a little bit more complicated if you want a brand new chemical entity that will do something that other compounds have not done before. But in general, they're very rarely disease modifying. It's very hard to find something that targets exactly where the mutation is and changes the origin of the disorder. If you're screening for and trying to repurpose, you don't always know how the small molecule is working, depending on how you're testing to look for the molecule. And it has on and off targets effects. So there are a lot of side effects that are associated with small molecules. The gene therapy advantage is it's a one-time therapy, but still we cannot redose. And people that have pre-existing antibodies to the virus cannot be dosed at all. So there's a barrier to treat certain patients, and definitely a big barrier to treat again in case you need a booster of the therapy. And antisense you heard a lot about, so I'm just going to skip. And if you have any questions to Jeff or me at the end, then feel free to ask us. So we know the target. What else do we need to know? So here I tried to give a little bit of a flavor of your child got a diagnosis, you got a diagnosis, you Google your gene, you look it up, you want to see what the protein's doing. And what do you need to know in order to go to someone and ask them to develop a therapy? Or what does that someone need to know? You need to know for gene therapy, it's really important to know what the size of the mRNA is, because that's what you're going to be putting into the virus, and that's what will make the protein when you're trying to replace it in your body. And not every mRNA fits in a cassette that goes into a virus. For example, in Duchenne muscular dystrophy, the gene is huge. So the trick there was to make a mini gene. And this is what raises the question whether if you cut and truncate the protein and it's not the original protein, is it really acting the same as the original protein? And this will be the question for many of the genes that were truncated and put in as a mini gene. You need to know what the introns and exons are for the gene. You need to know how the expression is regulated, especially if you want to look at upregulation and you want to know how your gene is modulated. You need to look at microRNAs or long non-coding RNAs. Jeff mentioned NMD and exon skipping. So all of these things you probably can just feed into a computer, and that will tell you a lot of the regulatory elements that are associated with the gene that you want to restore its function. And you want to know homology across species, and we learned the hard way when you have a gene that's very homologous between mouse and human and monkey, and you express it in the mouse and you express it in the monkey, and you want to see if you restored expression, if the gene is very, very similar among species, it's very hard to tease out the new expression versus what the animal is expressing in the background. So a lot of times we actually recommend to groups to optimize the human sequence so can be detectable over the animal background. What type, and you look at the mutated proteins, how is it mutated? How is it inherited? Is it a de novo mutation? Is it brand new in the family? No one knew before that anyone else suffers in the family from this mutation or carries the gene. That's important because if it is inherited, you want to go back and look at the mode of inheritance and find other patients that are potentially affected in the family. You want to know if it's a loss of function or gain of function. That's really important when you're designing a gene therapy or an antisense oligonucleotide. You want to know is there a genotype-phenotype correlation? If it's just a missense mutation, does it behave differently from a nonsense mutation when there's no protein at all? Or are the mutations all the same and they give the same clinical phenotype? This is really important when you start thinking about enrolling in a clinical trial. What are you looking for? What patients will you enroll in the trial based on the mechanism of therapy that you have decided to pursue for this indication? And then the last one I mentioned already, do you really need to know the protein function? As I said, I would argue that you don't have to know exactly what it does, but it's nice to know it if obviously there is information about it. What is proof of concept? And this is something that we're asked a lot by patient advocacy groups who want to partner with industry. What does industry want to see? So obviously it's not a one size fits all. Ideally, you want to see in vitro data, and it could be from 1 dimension, 2 dimension, 2.5 dimensions, or 3 dimensions. And if there is a good mouse model, we'd like to see data from a mouse model. Many groups have several different mouse models. It has to be the one, obviously, that's the most suitable for the modality that you've chosen. Cells could be either patients or isogenic cells, and organoids are now becoming more and more common, and especially for certain disorders and certain tissues, the data from the organoids is becoming really key for advancing things further into the clinic. And then the mouse model, a lot of times we have questions about, oh, we have a mouse model, but it doesn't behave like the humans. Is that okay? Well, obviously, ideally, we'd like to see some behaviors that are shared, but we want to know that the drug is working. So if your child has seizures, but the mouse does not seize, but the mouse has a different behavioral abnormality, if you can restore that, that's good enough. Or if you can show that the gene therapy or the ASO actually restore the mRNA and the protein, that's good enough. In the right place, that's good enough. So don't get stuck on the fact that the mouse doesn't behave like the humans. And there's always a question of when do you treat the mice? Everybody does P0 because it's really, P0 is immediately after the mice are born and the babies are tiny, tiny, and their skull is very, very soft. So it's easy to inject into the skull. And people tend to do that. But a P0 mouse does not represent a newborn or a patient post-birth, a human post-birth. So a lot of times people get really start insisting on seeing data from a 3-week-old mouse or a 6-week-old mouse. Showing in P0 is a good start. If your drug does not work in P0, it's likely not going to work in a later age as well. And of course, we need biomarkers, and I encourage every group and every company that's working on disorders to start looking for biomarkers, because having an early indication that the drug is working is really helpful. So I wanted to give this example quickly of what we had when we in-licensed the program for a disorder called WORI. It's a very, very rare disorder. I think there are 200 patients known in the world, plus minus. The kids, a lot of them die. So the number probably is around 200 around steady state. It's inherited, it's an autosomal recessive, and the key feature of this disorder is very, very severe seizures, up to hundreds of seizures a day, and the kids end up passing away because of complications of the seizures. So when the PI came to us with work that was funded by the advocacy group, what he had at the time, and it's all been published since then, was a good mouse model that replicated some of the human features, an AV9 gene therapy under the SYN promoter. So the protein is expressed in neurons, which is where we need it to be expressed. And he was able to show that the gene therapy reverses all the symptoms that are seen in the mouse. So early mortality, failure to thrive, hypoglycemia, seizures, other things that were observed in the mouse were reversed by the gene therapy. And we thought to ourselves, there are only 200 patients in the world. That's true. However, this gene therapy looks very compelling and we have to get it to patients. The proof of concept package was very robust, and we decided to bring the program into MODSI. Quickly, a few words about patient groups. You heard from some really great groups in the morning, and obviously great examples for what patient advocacy groups are doing these days and how advanced and how sophisticated the work is. On the left, you can see Cure Duchenne, the Cystic Fibrosis Foundation, and more recently FAST, the Angelman organization, that have been very successful by funding therapies. They're able to see a return on their investment and become very, very rich and very affluent and very influential advocacy groups. I just learned last week at a dinner that the Cystic Fibrosis Foundation has more than $3 billion under management, which is crazy, right? Everyone's dream to have so much money to be able to spend on your disorder. On the right-hand side is just a sample of the new CNS disorder groups that we're working with, and there are tens of them. And they're all hoping to be as successful as the ones on the left. So on sorry if I confused my right and left, but on the left is FAST, Cystic Fibrosis and Cure Duchenne, and on the right are the new groups, and they're just aspiring to be as successful, but they don't have the resources, and this is where MODSI comes in and hopes to support these groups and help them get into clinical studies and prove that the drugs that they're making are actually working and could be commercialized. So our mouse showed a really, really, really great slide on the tools that their foundation has. And I built this pyramid of all the information that advocacy groups can actually generate to, first of all, have a strong basis for clinical development, and also to attract industry, because all these tools are already available and the industry partner does not have to spend money on registries or natural history studies or whatnot. So having all of this is really, really valuable. It costs money, true. So what would be the priorities? The priorities would be things that are maybe started with things that are less expensive, which is serving the community, understanding the disorder better, putting in place a disease concept model, which just says what the priorities of the community are, versus spending a lot of money on looking for biomarkers, for example. But if you have the resources and you are able to do it, then everything on the bottom of the pyramid here could be executed by patient advocacy groups. As I said, registries, natural history studies, disease concept models, starting to think about clinical endpoints and the clinical endpoints that are meaningful in case your disorder has never been in clinical trials before, and in case you have some unique features and unique clinical symptoms that haven't been measured before, helping the industry partners and regulators understand what these endpoints are and how they're measures is really important. And I think that patient advocacy groups have are best equipped to actually design those clinical endpoints. Looking for biomarkers, is there anything in the blood, in the CSF, in urine that could be any imaging or EEG biomarker that could be adopted for early response to therapy? That's very helpful. And then if you go up the pyramid, looking at what Armel showed in her slide earlier today, having cells, having organoids, having a biobank that also came up this morning, and having animal models, the more the ones that could be used for drug development, the better. And then on the right-hand side, I would say that's where industry comes in. We can guide proof of concept. What should it look like in vivo, in vitro? What are the IND enabling studies that are needed? What toxicology studies are needed? What would a clinical trial look like? How many patients will we enroll? What would be the endpoints? How many visits does the patient need to have in order to get the therapy and to measure the response to therapy? And then the choice of the modality will be based on the expertise of the company. So you'll go to Everyone or in Lorem or La Jolla Labs if you want an ASO, or you go to other companies for a gene therapy and discuss with them what is the most suitable for your gene and your genetic mutations. Clinical development assumes that many of the disorders that we're working on have had limited previous clinical trial exposure before, and that we would like ideally to work on disease-modifying therapies. So as I said before, small molecules not likely to be disease-modifying. ASOs, gene therapies, gene editing, of course, will likely be disease-modifying, and an intervention will change the fate of the patient and the trajectory of the disorder. The challenges are to validate novel endpoints or to modify known endpoints to be suitable for your disorder. The endpoint has to be sensitive enough to measure change. And sometimes when a population is very diverse, it's hard to find something that could measure across a population. The studies are small. In most cases, especially in gene therapy, we do not want a placebo or a sham control because it just doesn't make sense to bring a very fragile patient in and have them go through the whole procedure of injecting something into their brain or something into their liver, and then they're not getting anything, and then bringing them in again when they are eligible for treatment. So actually the regulatory agencies are more open today to running especially gene therapy or viral vector studies without a sham control or without a placebo. And then sometimes you have advisory boards and you hear from the patients one thing, from the clinicians a second thing, and from the regulatory agencies a third thing. How do you collate everything and make sure that you're going into the clinic with something that will satisfy the needs of the patients on one hand, but actually could be approved on the other hand? And again, biomarkers, that my head of regulatory brainwashed me with a biomarker part. Quickly on disease concept models, we start with caregiver surveys, interview families to understand what they would like to see changed. We go through a medical chart review to see what information is already available without needing to go into a prospective trial. We then interview the families again in a more qualitative manner. So go deep into interviews, understanding what a day in their life looks like. And based on all this information, we design a natural history study. And all this is put together to get a conceptual model that will guide us on what the clinical trial would look like and what the endpoints are. And all this is done in parallel. So nothing should be done isolated. All this should be parallel tracked, of course. So whatever you can do and jumpstart in the beginning, you're better off later on when you want to get into the clinic. And just going back to the Mazi slide, I wanted to show that the interaction between academics and family groups is critical. And I think it will become even more critical if less grants will be available from the NIH, or there will be restrictions on federal money. A lot of academics will be relying on patient advocacy group money to do work in their labs. And I hope this won't create more conflict of academics needing to publish and to get tenure while families really want to get drugs into the clinic and treat their loved ones. It's a joint effort. There should be alignment on goals and the same sense of urgency. And it's a process, and especially for young investigators that never worked with patient advocacy groups or families before, it is a shift in thinking. And families need to understand that usually when somebody went to academia to head a lab, developing a therapy was not the first thing that they had in mind when they signed up for the academic career. And the collaboration is based on one hand, the scientific expertise of the academics and everything that they bring to the table, with the acknowledgement of the fact that the advocacy groups bring a lot of expertise that otherwise they will not have access to, the academics will not have access to if they don't work hand in hand with the advocacy groups. So in summary, it takes a village to develop a therapy, and alignment, collaboration, synergy, however you want to call it, it's critical for this whole process. And we need solid biology, very smooth handovers between academics into biotech, with working hand in hand with the advocacy groups. So imagine a 4x100 race where somebody drops the baton and loses the race because of that. We don't want that to happen. We need to parallel track, so run everything as smoothly as we can at the same time, and collaborate. So thank you, and happy to answer any questions you may have.